Hi, my name is Boris Zaydliner. I'm oncologist from Rostov Regional Cancer Center, Rostov-on-Don, Russian Federation. We will consider some issues in cancer comorbidities and immunology. The scope of cancer care has broadened from treating the disease alone to managing cancer-related symptoms at different stages of disease trajectory, including mental disorders. Such disorders are also one of the key targets in poor prognosis when a priority of cancer care becomes the relief of suffering. Among the nosological forms of comorbid mental pathology in cancer patients, affective disorders, depression and anxiety predominate. According to the WHO, depression is expected to be the leading cause of disability by 2030, and it's the most significant contributor to the overall global burden of disease. Medically ill patients experience depression at rates 50%. This disorder has devastating consequences on treatment adherence, quality of life, morbidity and mortality. While depression in cancer patients is often unrecognized and undertreated, the prevalence reported in most studies is probably an underestimate. It's now clear that patients with care are in population at greater risk for the development of clinically relevant depressive syndromes, with overall higher prevalence rates than those observed in general population. At the same time, evidence suggests that an affective comorbidity can precipitate the cause of chronic inflammatory diseases. In the opinion of Sfortini et al., this implies a bidirectional link connecting cancer and depression. Many interactions occur between the immune, neural, and psychological systems. In physical illness, the immune system serves as a sensory organ, communicating with the brain through the secretion of cytokines, which have peptide structure and produced by dendritic cells, macrophages, and also in the gut, in adipose, and other tissues. The cytokines possess specific receptors which are recognized as crucial unit on intercellular mediating. These substances are released as part of the immune response and affect neural, neuroendocrine, behavioral functions. On the other hand, there were revealed association between high cytokine levels and psychological disturbances, particularly depression. Lower plasma concentration of serotonin 5-HT at high circulating tumor necrosis factor alpha, TNF-alpha, at baseline have also been associated with somatic symptoms of depression during IFN-alpha treatment. Inflammatory cytokines IL-1, IL-6, TNF-alpha, and others are produced both centrally and systemically following stress. Their levels are elevated in medical illness. Tissue destruction by surgery, chemotherapy, and radiotherapy leads to damage associated molecular patterns on damaged tissue, which bind to pectin recognition receptor on leukocytes, particularly macrophages, causing the expression of the transcription factor kappa beta, NF-kappa beta, and the production of numerous pro-inflammatory cytokines, including interleukin-1, interferon alpha, IL-6, and TNF-alpha. Ionizing radiation and some chemotherapeutic agents are also able to directly stimulate NF-kappa beta independently of tissue damage and further increase inflammatory mediator expression. Psychosocial stress in healthy patients has also been shown to induce NF-kappa beta expression. These substances released by peripheral immune cells and or various tissues except the brain to initiate local immune activation by several mechanisms, including passage through leaky regions in the blood-brain barrier at circumventricular organs. Second, active uptake mechanism of cytokines across the blood-brain barrier. Third, local actions at peripheral vagal nerve efferents that relay cytokine signals to relevant brain regions, including the nucleus of the salivary tract and hypothalamus, the so-called neural root, the rapid pathway. Fourth, activation of endothelial cells and perivascular macrophages in the central vasculature to produce local inflammatory mediators such as cytokines, chemokines, PGE2, and nitric oxide. And fifth, activated immune cells such as monocytes, macrophages, and T cells can be recruited from the periphery to the brain. Here, the cells functions synergies with autocrine activity of microglia, probably of astrocytes, oligodendrocytes, and to some extent of neurons. The cytokines activate local CNS inflammatory networks to produce both alterations in neurotransmitter function and peripheral signals amplification, including inflammatory signal transduction pathways, cytokine production, and PGE2 release. In the brain, endothelial cells and perivascular macrophages respond to circulating cytokines to induce expression of cyclooxygenase 2, COX-2, and release of PGE2. Following an acute inflammatory stimulus, increased CNS cytokines can confer protection to the brain, yet under conditions of chronic immune activation by elevated inflammatory cytokines, microglia can become a source of inflammatory mediators that influence brain neurotransmitter systems and neural integrity. The persistent alterations in neurotransmitter function can lead to the development of depression and other neuropsychiatric dysfunction. Brain cytokines affect the neurochemical systems that are implicated in the pathophysiology of depression. It needs to mention some observations. Depressed patients display an activation to the monocyte macrophage arm of the immune response. Clinical diseases with an inflammatory component are associated with a high prevalence of depression. Antidepressant drugs have anti-inflammatory properties and attenuate the behavioral effects of immune charge. Based on these findings, it's suggested that immune activation is involved in the etiology and symptomatology of depression due to a general medical condition. It's biologically plausible that inflammatory cytokines serve as mediators of both environmental and genetic factors that may trigger the development of depressive disorders. Innate immune activation during chronic medical illnesses, as characterized by elevations in inflammatory cytokines, may contribute to the high rates of depression and other neuropsychiatric, e.g. cognitive or psychomotor symptoms observed in medically ill populations. Within the brain, cytokines induce a depression-like behavioral syndrome, frequently called sickness behavior. It's characterized by decreased mood, anhedonia, reduced capacity to experience pleasure, diminished interest in the environment and daily activities, reduced libido, anorexia, reduced food intake, weight loss, hypersomnia, psychomotor retardation, fatigue, and diminished cognitive functioning. These signs and symptoms are supplemented with confusion, hyperactivity of hypothalamic, pituitary, adrenal, HPA axis, and glucocorticoid resistance. This cluster is accepted to attribute to the account of most serious disease of person. Similar symptoms accompany medical conditions that involve immune activation in humans. Sickness behavior is seen in patients with depression or cancer. In the latter case, such cluster may be the result of both the disease and the treatment. Sickness behavior as outcome of cytokine release may provide a framework to explain many cancer-related symptoms, including depression, cognitive impairment, cachexia, fatigue, and a component of pain perception. As to underlying pathogenetic components of bidirectional linked cancer depression, the data are rather scarce. Both monoaminergic neurotransmitter concept of depression mechanisms and the limits of therapy on the concept are well known. High prevalence of treatment-resistant depression suggests that other factors than monoamine depletion could contribute to the pathogenesis of depressive disorders. In recent studies, great interest has called forth the association of mood disorders with inflammation as a part of the non-specific immune response that takes place after any type of bodily injury. Patients who are resistant to the antidepressant treatment demonstrate higher concentrations of circulating cytokines and acute phase proteins than patients who respond to antidepressant treatment and are at even greater risk of morbidity and mortality in medical illness. The results of some works have permitted to suggest systemic depressive symptoms from activating pro-inflammatory cytokines. The association between elevated levels of pro-inflammatory cytokines and the maintenance, as well the evolution of depressive symptoms, have become a cornerstone for the inflammatory hypothesis of depression. Meta-analyses have in fact indicated that in the absence of infectious pathogens, peripheral concentration of pro-inflammatory cytokines, especially that of IL-6 and TNF-alpha, were higher in non-medicated individuals with depression than in non-depressed individuals. Variations of IL-1 beta in relation to depressive illnesses were less consistently established, likely because of the difficulty in detecting with the very low levels of this cytokine in human circulation. In addition to elevated pro-inflammatory levels, low several levels of the anti-inflammatory cytokine IL-10 were negatively correlated to depressive symptoms in drug-free depressed individuals, suggesting that a shift toward a pro-inflammatory state comprising variations of either of both pro and anti-inflammatory functioning may underlie depressive symptoms. Increasing evidence has gathered of the presence of immune dysregulation in both depression and cancer. As a consequence, it has been hypothesized that immune activation, inflammatory and in inflammation may play a key role in the pathology of both diseases. The inflammatory pathway to depression was suggested. Such pathway appears to partially involve the communication between peripheral and central immune system. In fact, peripheral inflammatory cytokines have been shown to affect the brain through different ways. This finally results in the activation of CNS immune cells like microglia, the brain-resident macrophages. In turn, activated microglia promote central inflammation, which affects neuroplasticity, neurogenesis, and neurotransmitter metabolism, leading to behavioral changes typical of depression. Thus, cancer could lead to the development of depressive symptoms by inducing inflammatory responses and acting on the immune signaling to the brain. As well, immunotherapy with the pro-inflammatory cytokine IFN-alpha for some types of cancer promote depressive features. The latter findings not only support the view that inflammatory factors could play a provocative role in depression, but it appeared that the depressive effects of IFN-alpha could be attenuated by antidepressant treatments. The activations of pro-inflammatory processes directly or indirectly influences depressive states. Inflammatory cytokines released during medical illness can affect neurotransmitter systems that are involved in the pathophysiology of depression, including monoamines, glutamate, neuropeptide, e.g. corticotropin-releasing hormone, CRH, and growth factors, e.g. brain-derived neurotrophic factor BDNF, which is K for neurogenesis. IFN-alpha and other inflammatory cytokines can decrease systemic BDNF levels in humans. Inflammatory cytokines also influence BDNF receptor phosphorylation, thereby further interfering with BDNF signaling. Such divergences may adversely influence neurogenesis and neuroplasticity. Low levels of BDNF and neurogenesis have been implicated to the pathogenesis of depressive disorders. The 5H system is undoubtedly one of the most studied neurotransmitter systems in depression. 5-HT and other monoamines are released in the hypothalamus to mediate fever and early behavioral changes associated with sickness behavior. The role of biological substrate by which cytokines affect 5-HT neurotransmission and subsequently behavior may belong to the 5-HT transporter, 5-HT. Inflammatory cytokines, including IL-6, have been shown to increase 5-HT and noradrenaline NA expression and function. These cytokines and their signal transduction pathway, such as P38 mitogen activated protein kinases, MAPK, may increase expression of function of 5-HT and dopamine transporter. This effectively lowers synaptic concentration of 5-HT and NA and can lead to depressive behaviors. Inflammatory cytokines may increase 5-HT turnover in brain regions such as cortex and nucleus accumbens. These changes are congruent with the appearance of later, more persistent depressive-like behaviors. The low-grade chronic inflammation and pro-inflammatory cytokines, particularly TNF-alpha, have been shown to increase the activity of an enzyme which degrades tryptophan, TRI. TRI destruction results in reduced 5-HT levels. It's well established that TRI levels are decreased relative to monoamine levels in the brains of depressed patients. These interrelations will be presented in specific section. Taken together, these data suggest that innate immune cytokines can lead to a double hit on the synaptic availability of relevant neurotransmitters, influencing both their synthesis and reuptake, and thereby potentially contributing to the development of behavioral changes. Appealing to the clinical display of depression, we are invariably seeing both anhedonia and decrease of psychomotor activity on the top of manifestation list, both features being associated with dopamine GA functions. Their neurons play an important role in multiple mood disorders. Impaired GAergic neurotransmission may contribute to major depression in humans. One mechanism that may lead to basal ganglia changes in depression is inflammation. Inflammatory cytokines may affect multiple aspects of DA neurotransmission, leading to decreased synthesis, impaired packaging, and decreased D2R signaling, all of which may interact to a greater or lesser extent to reduce DA function. Positron emission tomography studies have revealed increasing uptake and decreased turnover of the radiolabeled TA precursor in the caudate and putamen of IFN alpha treated patients, which correlated with depressive symptoms. IFN alpha also results in decreased conversion of phenylalanine to tyrosine, which causes reduced downstream formation of DA in the brain and potentially depressive symptoms. This substance decreases the expressions of DA2 receptors and reduces striatal DA release, leading to feelings of anhedonia, behavior classically associated with DA release and reward. The peripheral inflammatory cytokines may specifically target the basal ganglia and DA to mediate depressive symptoms. Basal ganglia ventrostriatal activation to hedonic reward decreased during IFN alpha administration that correlated with reduced motivation. These alterations in ganglia function are possibly reflective of decreased DA availability that in turn contributes to the development of depressive symptoms. The changes of DA synthesis, release, and or receptor signaling have been proposed as potential mechanisms that may contribute to anhedonic and psychomotor symptoms. The K component of cytokine-induced depression is fatigue, that are thought to represent cytokine effects on the basal ganglia and DA function. In light of the role of basal ganglia DA in the regulation of motivation and motor activity, cytokine-induced changes in DA neurotransmission may mediate neurovegetative aspects of depression such as anhedonia, psychomotor retardation, and fatigue. 5-HT has known interactions with striatal DA and is thought to contribute to symptoms of cytokine-induced depression. Synthesis of DA and other monoamines is also dependent on the enzyme cofactor tetrahydrobiopterin BH4, which is necessary for both conversion of phenylalanine to tyrosine, the primary amino acid precursor of DA, and for tyrosine conversion to LDOPA. The sustained inflammation can increase both reactive oxygen species and inducible nitric oxide synthase activity that may ultimately lead to decreased BH4 availability and thus reduce DA synthesis. A key role of kynurenine in the link between cancer and depression was delineated by Fossini et al. Kine is catabolic product of three essential amino acids, which is the chemical precursor to the 5-HT and the hormone melatonin in both brain and other tissues. Meanwhile, in mammalian cells, the major route for tree catabolism is the kine pathway. The first step of the process is catalyzed by two enzymes, 3,2,3-deoxygenase TDO and indolemin 2,3-deoxygenase IDO. The latter seems to be an important link between the kine pathway and inflammation because of its inactivation by the innate pro-inflammatory cytokines including IFN alpha and IL-6. In the brain, IDO induction contributes to depletion and 5-HT synthesis decrease, and are believed to mediate the cytokine depression and corresponds to the expression of some depressive-like behaviors. As it's quite clear that tree metabolism is critical in both depression and cancer, the assumption that in patients suffering from various types of cancer, the development of depression might be associated to immune activation, especially to immune-mediated IDO activation, has gained more and more attention. Breitbart et al. Have studied patients with pancreatic cancer, which is known to have a particularly high prevalence rate of depression. In the tissue specimens of resected tumors, they've detected markedly elevated levels of IDO and TDO. It has been hypothesized that kine pathway is a biological basis for development of depression in this specific malignancy. Thus, the production of kynurenines seems to be involved in the pathway between pancreatic cancer and depression. Cytokine stimulate IDO promote the release of neurotoxic metabolites including quinolinic acid, queen, 3-hydroxykynurenine, and cause oxidative stress culminating in depression. Queen, following step in this metabolic cascade, is a potent and metal D-aspartate NMDA receptor agonist and is able to induce lipid peroxidation in urine that brings to excitotoxicity, whilst kine has been demonstrated to be anxiogenic. In severe depression, levels of quine have been observed to be raised in several regions throughout the anterior cingulate cortex. Neurotoxicity and degeneration, particularly on the hippocampus, have been implicated in the development of depression. This neurotoxic impact is counteracted with kynurenic acid, kyna, the next stop of the kine pathway. So there is a balance between neurodegenerative and neuroprotective effects of kine pathway expressed by the quine-kine ratio, which is strictly related to immune activation. This process involves the microglia and the astrocyte cells, which are the primary site for the tree catabolism in CNS. Kine is mainly produced by astrocytes, while microglia are responsible for quine production. The inflammation significantly shifts tree metabolism to kine production. An increased kine pathway activity may therefore be associated with various clinical conditions linked to immune activation, such as cancer and depression. The inflammatory patterns, including abnormalities of the kine pathway, may be implicated in the pathophysiology of depression. In a recent meta-analysis, the levels of these substances were compared in healthy persons and depressed patients. It proved to be the latter had lower kine values than in control sample, whereas on quine no difference was found, suggesting an imbalance of kine metabolites towards neurodegenerative effects. Therefore, antidepressants as well as other treatments such as omega-3 fatty acids could exert their antidepressive activity by reducing kine pathway-related neurotoxicity. Fatty acids opposing effects on inflammatory signaling are noteworthy. Omega-3 fatty acids have anti-inflammatory properties and are metabolized to form resolvins and protectins. These can influence cytokine synthesis and resolve inflammation. Conversely, arachidonic omega-6 fatty acid is both in intracellular second messenger and is also a substrate for the synthesis of prostacyclins and thromboxanes, which can stimulate inflammatory activity. In cancer, clinical presentation superimposes onto encountered immune activation that has been associated with relevant behavioral symptoms such as depression, and the kine pathway appears to be strictly implicated to this association. There is vast evidence linking clinical conditions characterized by a chronic immune response to the activation of the kine pathway. In some studies, both no correlation and negative correlation between kine pathway activity and depression were detected. These unexpected results put down to clinical variables and confounders, e.g. age, gender, metabolic disorders, as well as illness severity, antidepressant treatment. Interest to glutamate's contribution to depressive disorders was refreshed with data on the rapid antidepressive efficacy on NMDA receptor antagonists, e.g. ketamine. Kine-related glutamate release reduction was detected. Further, the A release, as it turned down, is in part under glutamatergic control. Therefore, kine may exert behavioral effects through interaction between glutamate and DA neurotransmitter systems, even in the absence of increased glutamate activity. On the other hand, quine contributes to oxidative stress and can directly activate the NMDA receptor. Quine and inflammatory cytokines have also been shown to increase glutamate release and decrease the astrocytic uptake of glutamate by the excitatory amino acid transporter. This combined effect of increased oxidative stress and NMDA activity contributes to quine-mediated excitotoxicity. Quine disturbs the glutamatergic transmission and has been implicated in a number of neurodegenerative diseases, including Huntington and Alzheimer's diseases. So, cytokines influence the glutamatergic system by a variety of routes. Such influences precipitate risk for depression. As noted above, the CRH secretion and release also increases with pro-inflammatory cytokines. The hormone is a key regulator of HPA axis, which plays a primary role in the negative regulation of inflammatory responses, along with glucocorticoids. Indeed, glucocorticoids such as cortisol are the most potent anti-inflammatory hormones in the body. These effects are largely mediated by protein-protein interactions between glucocorticoid receptor and relevant inflammatory signaling molecules, including NF-kappa beta. Thus, disruption of glucocorticoid signaling either through altered release of glucocorticoid hormones, including changes in the circadian cortisol rhythm, or disruption of glucocorticoid receptor function may contribute to increase inflammatory responses. Besides, glucocorticoid receptor disorders may result in the inability to shut down cortisol production, non-suppression, following administration of the synthetic glucocorticoid dexamethasone as manifested by abnormal dexamethasone suppression test (DST). Of note, abnormal DST responses have been associated with increased production of IL-1 by peripheral blood mononuclear cells in healthy subjects with major depression. Of relevance to the potential role of cytokines in this process, cytokine signaling pathway including P38, MAPK have been shown to disrupt glucocorticoid receptor signaling and thereby may contribute to reduced sensitivity of immune cells to the anti-inflammatory effects of glucocorticoids. Disruption of glucocorticoid receptor function may also contribute to altered HPA axis function. Sensitization of CRH pathways in the brain may induce an exaggerated response in adrenocorticotropic hormone and cortisol to the initial injection of IFN-alpha. Such inflammatory stimulus confers vulnerability to cytokine-induced depression. Drugs that enhance glucocorticoid-mediated negative feedback of CRH pathways, including phosphodiesterase type 4 inhibitors, through facilitations of glucocorticoid receptor function, may control CRH overexpression. Thus, the advantage of additional inhibiting inflammatory pathways is achieved. Kine pathways' roles in oncology isn't limited with comorbid disorders. Tree metabolism is also critical for cell proliferation and immune regulation. What's still unclear is whether the kine pathway in cancer is activated by the biology of the disease or simply by the related stress. This pathway's contribution to the tumorigenesis earns the particular interest. There is a well-known concept of immune privilege. In certain parts of human body, e.g. brain, eye, testes, fetal-maternal interface of the placenta, merely with a decreased reactivity to allergenic non-self antigens was detected. Such immune tolerance prevents fetal rejection and immune responses against immunogenic sperms. Next group unites body's tissues where expression of TDO, IDO1 and 2 becomes enhanced with the subsequent accumulation of tree metabolites. It consists of the placenta, the epididymis, maternal and embryonic tissues in early embryogenesis. Similarity of two ensembles is quite apparent and suggests these enzymes to play an important role in immune tolerance. Immune tolerizing effects are also observed in the local tumor microenvironment. An enhanced tree catabolism via kine pathway seems to be involved in immune paralysis against tumor cells. This may be primarily mediated by increased IDO expression and subsequent accumulation of tree metabolites, since IDO is either expressed by many tumor cells themselves or by tumor-associated cells such as dendritic or endothelial cells. When malignant cells begin to proliferate, the immune system's first response is local inflammation-induced tree breakdown. Meanwhile, the amino acid is essential for the growth and proliferation of all kinds of cells, particularly neoplastic. Later on, the tumor cells seem to develop countermeasures through both stimulation of IDO and tree availability limit for immune surveillance. Topical mechanism suggested if through the enzyme endolemin oxidase, which converts tree to kine intermediates. This is considered to be one of the central mechanisms underpinning the capabilities of tolerizing dendritic cells. Anti-tumor immune reactivity is also expressed in many cases by higher levels of tumor-infiltrating regulated T cells. It's suggested that IDO takes different positions in the three phases of cancer immunoediting: elimination, equilibrium, and escape. In the first phase, most tumor cells are recognized and destroyed by the immune system. Low-level IDO production in the tumor microenvironment contributes to this tumor defense by inhibiting tumor proliferation. During the second phase, heterogeneity and genetic instability progress in tumor cells that survive the elimination phase, thus enabling tumor cells to resist the immune response. In the last phase, escape, the tumor cells themselves, as well as tolerogenic immune cells, produce large quantities of IDO, which results in immune tolerance. IDO activity is strictly related to two key inflammatory cytokines, IFN gamma and IL-6, whose function is an inflammation and in cancer is widely known even if not completely understood. The first one has a prevalent anti-tumoral action. The second one is mainly a pro-tumorigenic factor. The sustained systemic immune activation in patients with malignant diseases is linked to tumor progression and a poor clinical outcome. It shows immune surveillance is a crucial component of cancer prevention. IFN gamma activates IDO expression, which in turn activates IL-6, creating a network where IDO acts like a negative feedback of IFN gamma and partially upregulates IL-6. As a regulatory interface between IFN gamma and IL-6, IDO promotes a pro-inflammatory response, contributing malignancy neovascularization. This enzyme seems to promote tumor progression by predominantly acting on regulatory T cells, effector cytotoxic CD8 plus T cells, and natural killer NK cells. These lymphocytes exert a protective activity by reducing tumor progression and improving anti-tumor immunity, but can suppress CD8 plus T and NK cells. It induces apoptosis of these cells, probably mediated by oxygen-free radicals, while 3HAA queen precursor induces apoptosis of monocytes macrophages. Nearly all metabolites of KIND pathway affect immune activity via several mechanisms. Tri-depletion slows down protein biosynthesis in immune cells and induces cell cycle arrest of T cells. In addition, the final product of the KIND pathway, NAD+, also induces apoptosis of CD4 plus and CD8 plus lymphocytes. Apart from immune modulating properties, KIND metabolites may also help tumors to optimize their microenvironment. Formation of queen by glioma cells was described to promote resistance to oxidative stress. Additionally, tumor cells may enhance their own IDO activity. On the other hand, 5-methoxytryptophan 5-MTP, which is produced by mesenchymal cells such as fibroblasts, inhibits migration of cancer cells, tumor growth, and cancer metastasis. This effect is probably mediated by 5-MTP-derived inhibition of COX-2, which is constitutively overexpressed in cancer cells and promotes cancerogenesis. Therefore, reduced 5-MTP formation due to decreased TRE availability can contribute to tumor growth and cancer metastasis. Plasma biomarkers of inflammation and KIND pathway activity are independent predictors of mortality due to cancer, and the latter can be used as a prognostic factor. In particular, even at the early stage of cancer, IDO activity is enhanced, and such activity in the vast majority of studies has been associated with a poor prognosis. Moreover, IDO activation may be linked to the development of cancer-related fatigue and thus to its debilitating consequences. Given IDO's crucial role in tumorigenesis and progression, molecules with the ability to inhibit IDO activity have increasingly gained attention in immuno-oncology. This inhibition may be advantageous to improve response rates to anti-cancer immunotherapies such as checkpoint inhibitors. Immune checkpoints are molecules that regulate the immune system by modulating immune tolerance and preventing uncontrolled immune responses. Specific checkpoints inhibitors are therefore emerging as new pharmacological targets to enhance cancer treatment. An important limit for this new treatment strategy, however, is that cancer can still maintain an immunosuppressive microenvironment by activating IDO and the KIND pathway. Thus, cytokines may have an important role in explaining the high rates of depression often observed among cancer patients. The depressive symptoms in these patients can be precipitated by somatic factors. The KIND pathway is one of the biological links between depression and cancer. The clinical implication is that the mood disorders in cancer patients may arise from biological stresses, which is an important message to help destigmatize the cancer-related depression. However, most studies are required to further analyze the role of trimetabolism in the development of mood comorbidity in oncopathology. Thank you for attention.